Hello, everyone, and welcome to today's live broadcast, Beyond 100,000 Exomes, Insights and Lessons from Large-Scale Sequencing in the Cloud, presented by Dr. Jeffrey Reid, Executive Director and Head of Genome Informatics at the Regeneron Genetics Center. We are excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. This webinar is sponsored by DNA Nexus, the global leader in genome informatics, data integration, and collaboration. The secure, scalable, and collaborative DNA Nexus platform helps researchers across a spectrum of industries, pharmaceutical, bioagricultural, clinical diagnostic, government, and research consortia accelerate their genomics programs globally. I'm David Shaywitz, Chief Medical Officer at DNA Nexus, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located on the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window, or use the Q&A button to let us know that you're having a problem. Also, this is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located in the bottom left corner of your web page and follow the process for obtaining your credits. Now for the good part. I get to introduce Dr. Jeffrey Reid, Executive Director and Head of Genome Informatics at the Regeneron Genetics Center, who has quite the impressive track record and who I've loved working with and who's taught me almost as much about genetics as he has about the right way to make classic cocktails. For over 6 years, Jeff served as the leader of the Next Generation Sequencing Informatics Group at Baylor College of Medicine before joining Regeneron. In Tarrytown, Jeff has helped Regeneron build one of the world's most comprehensive genetic databases, pairing the sequenced exomes and de-identified electronic health records of more than 100,000 people to date, with the audacious goal to ultimately sequence more than 250,000 people. This pioneering genomic sequencing study, which has profoundly accelerated target ID at Regeneron and impacted patients at Geisinger, was made possible by Jeff's intuitive appreciation for the power and utility of the cloud and of the DNA Nexus platform, which has enabled and catalyzed a range of research partnerships through the sharing and management of large-scale sequencing data and tools. Please join me in welcoming the always stylish Dr. Jeff Reid. I will now turn the presentation over to him.
Thank you, David, for the kind words and for the introduction, and thanks everyone for being here. So what I wanted to talk about today was some of the really exciting and interesting things that are happening in genetics and genomics, and particularly what we're doing at the Regeneron Genetics Center now that we've sequenced more than 100,000 exomes with a variety of partners. So let me start by just giving you an outline of what I want to talk about today. I'll start with a little bit on the vision of the Regeneron Genetics Center, talk some, maybe a little too much, but on automation and informatics and the things that are really the infrastructure pieces that are fundamentally important to enabling the kind of large-scale sequencing that we're trying to do here. And then get to the really good stuff, talk about some of the biological insights that we've gained from some of the studies that we've done, and really get at some of what has now become recently possible with large-scale genome sequencing and computation. And at the very end, I'll talk a bit about the future. Oh, I do want to mention in the lower right, you'll see where the data or some of the effort has been expended in a collaborative way. Since we're a highly collaborative institution, I like to give a little shout out to our collaborators. So if you see someone's logo or name in the lower right corner, this is really work that's shared between us and them. So starting with the RGC vision. So what is Regeneron? We're a large cap biopharmaceutical company. We were founded in New York City in 1988, And on the right, you see our two of our founders, Len Schleifer, who is currently the CEO, and George Yankopoulos, the Chief Scientific Officer, and they're still here at the company, which is quite exceptional and is one of the key resources within the company that allows us to keep a focus on technological innovation and scientific excellence. We've received a variety of awards. I'll point to recently we were #3 on the Forbes list of most innovative companies, and this is quite different, as I've learned coming from academia into the pharma industry. This is quite different than how many pharma companies operate, and really fundamentally important for the things that we're doing at the RGC, because Regeneron as a company has built a functional mouse genetics effort that is world leading, and pairing together with that the idea of building essentially an academic genome center embedded in a pharma company is what is enabling the discovery genetics and the development of new and better therapeutics with the leadership of people like Eris Barris, the head of the RGC. You know, we really believe that if you're not exploring human genetics at a fundamental level, you're not really doing modern day drug discovery. And that being made real at Regeneron through the RGC and our functional mouse work that I'll talk in detail a little bit about as I get farther along. But you might ask, why genetics? There are a variety of different ways of trying to innovate within healthcare, and really I'll point to some results that were published a few years ago that with genetic evidence, drugs succeed at a much higher rate. So as some of you I'm sure aware, there is a catastrophe in terms of drug failure in the industry. More than 90% of drugs that enter phase 1 clinical trials will fail. And in phase 2, as you know, you move farther along the pipeline, things get more and more expensive. You have a lot of failure from lack of efficacy and a lot of failure from toxicity. And the hypothesis here is that a lot of this failure comes from the fact that the preclinical models are poor predictors of benefit. Curing a disease in a mouse is not always the same as curing a disease in a person. And so by having genetic evidence, being able to really understand the impact of a therapeutic on a living person by having a genetic proxy gives you a much higher chance of having a drug succeed, and in the end both create something that helps patients and creates value for the company. So we feel at the Regeneron Genetics Center that genetics is the single best path to improving the outcomes for patients and improving the success of the drug development process. So this is really what has driven the pairing of a human genetics effort, namely the Regeneron Genetics Center, with Regeneron's history of great mouse genetics in our VelociGene platform that allows us to humanize mice and do various functional studies in the mouse in a rapid and accurate way. And really the discovery process now, by pairing the human genetics and the mouse genetics, allows us to have a virtuous cycle of feedback where we can maybe identify a new target based on finding an association between a genotype and a phenotype in a genetics study, use further data to try to understand what biomarkers may confer response or non-response based on a therapeutic, look at new indications for drugs that are currently in development or currently in the market, identifying ways that drugs that are already approved can help additional patients, and then de-risk targets, so identify so-called on-target adverse side effects, things that are coming along together with the benefit of a drug that might be damaging, by looking for people who have, for example, loss-of-function or gain-of-function variants that confer a sort of humanize or confer a copy, if you will, of what the drug would do. So to try to deliver all of these insights at the RGC, we are playing across the full spectrum of genetics studies. We have general population study, our foundational study that I'll talk a lot about, with the Geisinger healthcare system, the Discover cohort sequencing, which allows us to identify a broad spectrum of people, get deep electronic health record data from which we can mine for a variety of phenotypes, and looking at very large sets of individuals, identify both common and rare variants that we can do statistical testing. Also, at the other end of the genetic spectrum, we have a variety of collaborations around family studies where we can identify individual families with carriers of high impact variants that cause high penetrant disease. There's a variety of collaborators, ever-growing list, including places like Columbia University Medical Center and Toronto Sick Kids Hospital, who have spent years and years collecting various samples across a whole spectrum of phenotypes. And this is really kind of where a lot of the nuts and bolts genetics can get done, and feeding back into our insights about genetics by looking for the same variants that we're finding in family studies in the general population. Somewhat in between the two, we can look into founder populations. Basically, these are populations that are like extended kindreds or extended families that have gone through some kind of bottleneck where the frequency of particular rare variants are much higher in these populations than one would normally expect because of their founder the effect of founders in the populations, and this allows us to do kind of both family studies and general population studies at the same time, collecting large numbers of people who share rare variants, but also finding a high penetrant disease and really chasing down the impact of a particular variant and even looking in some cases for modifiers. Also, we have an ever-growing list of collaborations around phenotype-specific studies where we know we have an interest in a particular phenotype, and we will work with leaders in the field within those phenotypic areas to try to identify people who have disease who can be helped, where we can help both understand their disease and maybe help impact disease through the development of therapeutics. So, talking about this foundational collaboration I alluded to earlier, when the Regeneron Genetics Center launched in 2014, we knew we needed a really great partner to launch something really, really big. Because as I'll convince you throughout this presentation, I think, there is the unique value of having large numbers of individuals so we can collect large numbers of carriers of rare variation. So the concept of the launch of the RGC with the Geisinger collaboration was really to build the world's most comprehensive genotype-phenotype resource, combining de-identified genomic and clinical data for more than 250,000 people. This would aid for our part in the drug development efforts and are trying to understand biology and disease better, and for Geisinger's part, it gives them research data that they can do clinical validation on and report these data back to patients so patients can really try to improve their care and really deliver on some of the promise of personalized medicine. Now, Geisinger is a phenomenal healthcare system. They have 2.5 million patients, roughly, with cradle-to-grave records, which are quite rare. There are very few healthcare systems that adopted the electronic health record system and really deployed it effectively and maintained it over years and years and years, which has really put Geisinger into leadership in clinical informatics and makes them an ideal partner for us. So if you're not familiar with Geisinger, this is a picture of the main hospital. On the right, you see a map of Pennsylvania. This is, I believe, quite old. They've actually expanded since this slide was generated. But the point is, geographically, they are primarily in the central Pennsylvania area, although extending out into outlying areas. And one of the interesting things is because this population is so well captured by the healthcare system, we get individuals in extended families, and we also have individuals staying in the population for a long time, which really makes Geisinger one of the world's largest longitudinal live populations, where we can see patients again and again and again, and as we understand more about their disease, that can be fed back to them, and we can do a variety of callback studies and really try to get at the biology of individuals and leverage that understanding of the biology of individuals to more generically understand better disease. So that's all I'm going to say in terms of introduction of the Regeneron Genetics Center and our foundational collaborations with Geisinger. Now I want to pivot a little bit and talk some about what enables this under the hood. And let me just start with the idea of collaboration. One of the things that has been a major enabler of the Regeneron Genetics Center is not just our collaborators externally, but our internal collaborations with a variety of groups. And you'll see this in this slide. We have an unparalleled automation team at Regeneron that has helped John Overton and his team in the sequencing and lab operations group at the RGC build out probably what is the single most optimized automated process for exome sequencing in the world. We have an automated biobank. I like to call it a vending machine for samples. So you just put your samples in, you type in the number of the sample, or you give it a spreadsheet, and it aliquots the samples back out for you. We have library prep automation. It's not quite walk-away automation, but there's very little human intervention that allows us to prep more than 200,000 samples a year. This feeds into what is currently a fleet of Illumina HiSeqs, although recently Illumina announced that we are one of their early partners in the NovaSeq platform, so we are continuing to innovate and stay at the forefront of the sequencing technology as well to try to push into ever higher sequencing rate. Currently, we're at the order of between 100 and 200,000 exomes a year there. And then in terms of the cloud-based informatics, which I'll talk quite a bit about, really, that has been a huge enabler for us in terms of the rapid scale-up. We've done some scale testing. We know that the system, as we have it in place now, will bear at least 500,000 exomes a year, and we're quite comfortable with moving into 1 million and beyond when we can get there in terms of sample flow and sequencing. I do want to point out that we were really one of the first genome centers to fully commit to the cloud. When I started at Regeneron, this was what our server room looked like. This is, we used to give a tour, and I had a little photo of a cloud on the window that you can see there. When we took the tours through to show them where we would have been putting our data center, the room actually said data center on the door, and we, with an idea from one of our founders, decided it would be fun to get a cloud in the room, so we bought a fog machine, and we would actually make the cloud in the room until it set off the fire alarm. And so we don't have the fog machine anymore, but we still have this idea that having local compute is just not scalable enough, and it ends up being too expensive to maintain. So really, our compute architecture looks something like this. So in the blue box at the bottom, you see what is the RGC. We're essentially a collection of sequencing instrumentation, lab equipment, and then laptops, desktops, and workstations. We have a few servers here and there, but for the bulk of our compute, we can either go over two 10 gig direct connects into AWS or over a 1 gig corporate connection through the open internet into AWS, where our primary platform partner is DNA Nexus. And what we do with them is we really have two areas. So we have the platform area, highly secured. It is allows for extreme automation. So it's the thing that has been hugely helpful in our building out the computational automation for data production, and that's where all of our production and analytical workflows rest, as well as our data sharing profiles. So this is how we very easily share data with our external partners. We can put data in common areas and share it securely, and they can compute on it, download it, or do whatever they like with it. We also, of course, as a science-focused and innovative genome sequencing effort, like to kind of do new things and try to do things that a bit differently, maybe that don't fit in the production or analytical workflow framework. And so we have a separate area, we like to call it the annex, which really allows us to provision secure virtual data centers. And this is really a great solution for everyone in that the enterprise IT group still has oversight and policy management, so they're happy. It still has the security that everyone wants and demands, and it gives us the flexibility to then try new things, spin up new types of clusters, spin up instances that do a variety of types of analysis, and keep everything under control and secure through continuous guardrails. So this has been hugely effective in helping us go from a genome sequencing effort idea in early 2014 to a sequencing center that's produced well over 100,000 samples by the end of 2016. So really, you can think of us as a collaborative exome genotype factory where we have this cloud-based hands-free automation that goes essentially from raw data to sample-level VCFs. I do want to, in the spirit of that collaboration, point out that our LIMS team and our IT teams have been fundamentally important in all aspects of data production here. So we have integrated QC outputs from the production pipeline, which may seem a little bit boring and pedestrian, just making sure that the data is good, but it's incredibly important from the business perspective of making sure that you're making good data effectively. But there's also science that can really be done with this data. I'll get into some detail about this when I talk a little bit about what we've done with copy number calling, but the ability to gather a lot of QC data has been enabling in terms of our processing data and getting to understand variation in the individuals we've sequenced better. Our data deliveries look like, I like to call them data freezes, where we take a punctuation point in a project and roll up samples into project level VCFs accompanied with QC data and ancillary support files. Now, I do want to say a word about joint genotyping. For those in the field who think a lot about this, there's a question about what is the value of calling variants in samples in the aggregate, taking many samples together, and calling variants essentially across all samples, or calling samples in individually or in blocks or bundles. And what we know is if you try to joint genotype hundreds of thousands of samples at the same time, it gets very time-consuming and expensive. So one of the things that we're working on with our collaborators at DNA Nexus is a better framework for doing this that Mike Lynn and his team are working on that they call GL Nexus. And the idea here is to essentially obviate the need for taking and doing this joint genotyping process at punctuation points and having the data be more continuously integrated, and then creating the opportunity to just do requests. So for example, just request genotypes within the gene ALDH2 for East Asians, and then query that data repository that can give you that data that's already been properly genotyped together. Or if you wanted to say, give me all of the data, the production data from Project X within a given time frame similarly. So we're looking forward to this and other innovations that are fundamentally enabled by cloud computing and our collaboration with some of our partners. But now to the data. So I'm going to talk primarily in this section about the first 50,000-ish, I like to call it freeze 50K, patients from the Discover collaboration, our collaboration with the Geisinger healthcare system, where we sequence the exomes of people who have been more or less agnostically gathered across a variety of clinics within Geisinger. So just to set your thinking about what one finds when one sequences the exomes of 50,000 people, in total we have about 4 million single nucleotide variant sites. So it's interesting to point out that the vast majority of variation that you find within the population is common. Common variants are carried by a large number of people, and so if you pick any random variant that you find in a pool of variants from a large number of individuals, that variant is very likely to be common. But we tend to account for variants in terms of sites. And in terms of sites, actually, as you sequence deeper and deeper into a population, you keep adding more and more sites, but those sites tend to be rare because you've already found the common variant sites. So the value of sequencing the 50,000th and 1th person is the additional rare variation that you identify in that individual, as well as having some value in accruing numbers of individuals who carry variants and having information about their phenotypes, which I'll get to. But this is fundamentally important to the strategy that we have, that as we're sequencing more and more into the larger population, we're actually accruing larger and larger sets of variation sites. And so I think this slide is a little bit interesting because what you see is at 50,000, actually a very small number of sites, about 100,000 out of 4 million, have an allele frequency above 1%. So most of the things that we're finding are rare. And that's interesting because the thing that is most interesting to us is loss of function variants, or as I like to say, putative loss of function variants, because we don't know for sure. We can algorithmically identify variants that are likely to impact function, but there are, you have to go through a quite rigorous process of functional validation to really understand the impact of any given variant. So this is an interesting statistic from our data that each participant harbors roughly 20 or so predicted rare loss of function variants. So this gives you some indication of how interesting any given individual might be from the perspective of accruing variants that are likely to have a significant impact on function. We think that these should be rare, of course, because if it was a variant that significantly impacted function, it would be selected against. We also anticipate that these things should not, individuals shouldn't be carrying enormous numbers of them because, of course, these are people who are in a healthcare system, and yes, they're seeing the doctor, but for the most part, they're walking and talking, and they're not people who are incredibly ill. So in your sort of average person, you can imagine that you'll find something like 20 putative loss-of-function variants that are rare. So this slide then speaks, I think, fundamentally to the strategy of doing large-scale sequencing. Because we know that most of the sites we're going to accrue as we sequence across lots of people are rare, and because we know that there's something like 20 possibly interesting rare loss-of-function variants per individual, what you see is if you want to accrue more than one individual who's a heterozygous loss-of-function carrier, well, we can do that, and if you look in terms of the number of autosomal genes where we have at least one or more individual carriers of heterozygous loss-of-function variants, we have 92% of the genes that we are looking at, of the 18,000 that are in our list, where we have at least one heterozygous individual across these 50,000 people. But of course, it's hard to do statistics. It's actually not really statistics with one person. That's more of an anecdote. So if you want statistics, you want to get into 5 and 10 and 20 carriers, and maybe you're interested in complete knockouts. Maybe you're interested in homozygous loss-of-function carriers. So you can see in this table that if you want to get into the statistics of total knockouts, if you want 10 individuals for each gene who are homozygous loss-of-function carriers, well, at 50,000 people, you've only ascertained enough individuals to do that kind of statistics in 1% of the genes. And so this points us to the idea that we think that we're going to need to continue to sequence very deeply, and this is the thinking that I think is driving many in the field to sequence very large cohorts. And you know, the statistic that I like to have in mind is, yes, 92% of genes with one or more heterozygous loss-of-function carrier for 50,000 people, but only about 7% of genes with homozygous loss-of-function. And this includes many Regeneron drug targets, which is already exciting for us, but if we really want to get into the whole of biology, we need lots and lots of people. Now that's just from the genotype side. Getting into the real value of the Geisinger healthcare system, and what's really exciting about what they've built over the last really 20 and 30 years is their data warehouse, basically, their database of people's healthcare data. So they have something like data on 1.5 million patients. It's refreshed monthly, at least for us. They are, you know, continuously, of course, working on it on their side, but we get a monthly update. This has something like 140 tables, and I don't expect you to read this slide, but this shows you somewhat the complexity of the interrelationship of this data. More than 2 billion rows, and this covers more than all aspects of really just every possible thing you could imagine. In addition to like the usual aspects of an electronic health record, there's many ancillary data sources that are integrated here. Unique data types like cardiac catheterization, pulmonary function testing, echocardiography, and really to set your mind about how large this data set is, it grows at a rate, in terms of the largest tables being lab results and vitals, it grows at a rate of from about 500,000 to 1 million rows per week, these individual tables. So this is really a living data set. This is something that grows on a daily basis and is just incredibly valuable in terms of being able to go back to patients and having large numbers of individuals with lots and lots of incredibly valuable data. Something like more than 400,000 patients with at least 10 years of longitudinal EMR data is just really unparalleled. And with enormously valuable and interesting deep dive data sets, things like coronary artery disease, COPD, bariatric traits, liver, things that are you know, high unmet need and really where we think we can have a lot of impact in terms of drug development and helping patients. So now that I've explained a little bit about the phenotype data and the genotype data, just a couple words on how we bring it all together. So this cartoon kind of shows the perspective. So we have a variant database that we're gathering from a variety of partners from both population-based studies and family-based and cohort studies. We have this unified phenotype database that's getting input, again, from these large studies with really great longitudinal data like Geisinger, as well as data sets that have less rich data, but nonetheless is valuable to include from some of the smaller studies. And then we can bring these two things together in the cloud and apply applications to do cross queries and cross analysis to them. So this is really the bread and butter of what we're doing. So we're taking these genotypes, we're taking these phenotypes, and then we're making a variety of queries. For example, focused hypothesis-driven queries. So are variants in a specific gene associated with a specific phenotype? So maybe we have a particular idea about an on-target efficacy effect for a known drug, and we want to test it genetically. We can do that. We can ask that specific question of this data. Phenotype first strategies. This is more in the novel drug discovery space, where we can say, okay, what genes have variants that are associated with this gene of interest, or this disease, or this trait of interest, and see maybe is that a druggable target? Is that a gene that we think might be something where we could plausibly look at putting together a development program to see if that could be a good lever for improving the outcomes for patients that have that disease. And also, really interesting with this data set because of the richness of the phenotype data, genotype first strategy. So saying what phenotypes in the health record is a given gene variant associated with? Can we identify, are there new indications for a drug target, or are there unanticipated adverse effects that can really inform a program as it's moving through development? And most excitingly is a lot of this is automated now for us as well. So we don't have to wait to think and ask the question. We can pre-compute some of these things, and then those questions can be asked in terms of queries on a data resource. Now, a little bit of a sideline. I want to get in. I had mentioned earlier this idea that the QC data can have some fundamental value beyond just understanding the QC. And so one of the stories I really wanted to hit was talking about expanding the scope of what we do at the Genetics Center here. So primarily what people historically have looked at are SNPs and indels. These are single nucleotide variants, small indel variants. But we know that copy number variants have a huge role to play in some particular phenotypes, some particularly tough phenotypes like autism, as well as in some cardiac phenotypes. And just in general, copy number variants have been incredibly under studied and under ascertained in a lot of large-scale studies because they're difficult. And so, the way they've been done in the past, you look at a SNP array or an array CGH, these platforms have limited probe density, and with that limited probe density comes limited resolution, so you're only seeing large things, you're only seeing things in regions where you know to look. You can do whole genome sequencing and really do a deep dive on structural variants. That's of course quite expensive, and it is generally only done in small studies, although I think it's becoming more and more common now. But one of the challenges from doing what we're trying to do here with exomes is calling CNVs from exomes is very challenging, because you're stuck looking for copy number variants in the context of noisy read coverage in these small exon windows where you've created a capture reagent to select out the regions of the genome you want. And in looking at the previous exome CNV callers for our needs, we're working at large scale, so we need scalable pipeline-ready solutions. We're interested in common disease and rare disease, so we need both rare variants and common variants. And we need to get CNVs down to high resolution, down to single exon resolution, ideally, and maybe even below. So we approached this problem with the idea that we could do something uniquely impactful and interesting here because we had a data set that had such great QC. And the reason that's important is the way our algorithm works is the single most important driver of variability in coverage is GC content. So the first step is you just do a normalized read coverage accounting for the local GC content. But the next step is after you've done that GC normalization, you need to do a further normalization. And the problem historically has been, how do you know what samples to include in that normalization? How do you best take out the variability of sequencing and leave in the variability of the genome? And for us, the aha moment came when we realized that we could essentially, using a clustering in QC space, identify samples that perform similarly on the sequencing platform, but maybe are completely dissimilar in terms of their genetics. So we could use samples that sequenced in a similar way, that performed in a similar way on the sequencer, to normalize each other, and then use that to gain better insight into what the copy number variation was from the genome. So after that QC kind of clustering step, we can then fit a mixture model. And I'll show you a cool cartoon example of that, where we can identify what the expected copy number state for a given individual in the distribution is, and then stitch those together with a hidden Markov model into contiguous exonic regions. So here's the cartoon of the way this works. If you're a person who is diploid genome and we've sampled your genome in this region of copy number 2, you could see a coverage distribution for a number of samples that looks something like this. And you can easily fit a unimodal distribution, a nice normal distribution there. But if you have a site that's variable, where some samples are copy number 0 and some samples are copy number 3, most are diploid, some are heterozygous with a single copy deleted, it's really not useful to fit that single distribution anymore. And this has been really a big confounder in trying to understand the impact of common copy number state, or the impact of common copy number variation in calling a copy number. But because we now can normalize our samples and we can stratify into these nice tight distributions, we can instead fit something like this, a mixture model, where we can really now get at, okay, for these particular samples in this cluster, they are very likely to be copy number 2, or very likely to be copy number 3. So showing some real data after this model fit, you can see first and foremost, these are different exons of the same gene. You can see first and foremost that the normalization actually can vary quite a bit from site to site, from exon to exon. But also you can see the quality of that fit. So the red dots are called copy number 0, the blue dots are called copy number 1, the black dots are the normal diploid state. And you can see clearly how with the multimodal fit, you can identify crisply, even at the borders of these things, which samples are carriers of copy number 1 and which are copy number 2. So to show you some real data, some validation data, we went through an exercise. Because we have a lot of related individuals, of course, in Geisinger, we're talking about a healthcare system, and families go to doctors, not individuals. And so we were able to collect data from 6,500 parent-child duos, and then we could look for what the expected transmission rate. If we could validate with genetics, the expectation if the parent has something, the child has a 50% chance of having the same thing. We can then use that as a validation on our data and compare it to some copy number data that was generated on genotype chips using the gold standard genotype chip CNV caller pen CNV. And what we found was maybe unexpected, but I think it was quite exciting to us, that we do actually a little bit better at the larger copy number state than the array. But once we get into the minimum size cutoff that's below, let's say, 10 kb or 3 kb, the data on the x-axis in the plot here, what you see is the yellow line is the pen CNV data, and it really falls off rapidly. It's not the fault of the technology, it's really the fault of the fact that the spacing of probes on a CNV array tend to be farther away, and so it gets really, really essentially impossible to ascertain copy number variance across large scale. Whereas with exon sequencing, because we're doing high resolution sequencing in the regions that we're targeting, we can actually maintain a transmission rate between 40 and 50% all the way across the spectrum of size. Now, I'll remind people, again, the transmission rate of 50% is the ideal, and so yeah, the calls do get better as you get larger, but still having a transmission rate above 40% at a scale of 100 base pairs is really, really exciting. And you don't have to believe the comparison between the pen CNV calls and the Clams calls, our CNV caller. We also did a separate independent validation on some loci using TACMAN, quantitative PCR. And so what this shows is at this particular site that we had 92% sensitivity and 98% specificity for small CNVs as validated by TACMAN with essentially a 47.6% of 50% transmission rate for rare heterozygous calls. So we have done validations now to convince ourselves that we can detect both common and rare copy number, validated then again back in the transmission. And so we think that with this data now, we have really one of the most exciting copy number data sets anywhere in the world. So with that, I'm going to quit talking about some of the kind of infrastructure and some of the details of how we got to the data set that we have. And I'm going to get really into what I think is the really exciting stuff about the biology. So let me just start with this figure from a paper that I really love. This is a paper from Jim Lutsky and others at Baylor talking about this concept of clan genomics. That is genomics that's across an extended pedigree of individuals. This is not, as many people work on the far right, in small families where you have relatively new variants with relatively large effect, with really high penetrant disorders, things that are very, very clearly genetic. It's not necessarily the things at the far left where you have lots and lots of variants of small effect. They're relatively old variants, and these studies are generally done in completely unrelated individuals. In fact, in many of these common complex disease studies, historically what you would do is you would try to ascertain individuals who were unrelated, and then if you accidentally found individuals who were related, you would actually exclude them from the study. So I want to talk about our results in the context of this concept, this idea of this spectrum from variants of large effect that are relatively new, acting in small, closely related families, and then relatively small effect variants that are quite old, that are acting in large, unrelated populations. So to start out, I'm going to start out on the far right of that plot. And I'm going to show you some results from our Mendelian disease analysis pipeline. So as of sometime last year, I don't remember exactly when this data was finalized, we've done something like 10,000 samples through our Mendelian pipeline. This is over 1,000 families, and what we found is really exciting. There are something like 99 families with known causative variants, about 120 families with novel variants and known disease genes, almost 150 novel candidate disease families where they have novel candidate disease genes, and 225 families with multiple candidate genes. And the way this works is you sequence the individual affected with the disease, the proband, and then you immediately filter that down to about 20,000 to 25,000 coding variants per individual. And then you go through an annotation and filtering exercise where you take out basically everything that you expect to not have an impact on disease in this way. So these are things that are synonymous. These are things that are incredibly frequent in large populations. These are things that are not well conserved or not thought to be deleterious, things that basically don't seem very likely to be a high penetrant variant that may be relatively new. And have strong effect. And then looking in trios where they're available, and in families we can identify potential de novo variants, compound heterozygous variants, and variants that segregate with the disease in families where there are affecteds and unaffecteds. And that brings us then ideally to a relatively small list of genes and variants where we think that they're very likely to be causative of the disease, and then we can look across multiple families. This is a methodology that a variety of people have applied. This is not new with us, although I think we do it quite well, and actually we have, again, with that theme of automation and collaboration, we've had a lot of the informatics teams working closely with the Mendelian disease teams to build out a lot of automation. And hopefully soon we'll be doing this on a decision support basis, really having the analytical pipelines be completely automated, and then the Mendelian disease expert doesn't spend a lot of time digging in on the actual munching around of the data, but spends their time looking at things like this. So this is some data that we made for the Columbia University Medical Center group, where they have about 71 families and 192 singletons with pulmonary arterial hypertension. And the idea here was to take a lot of individuals collected in these families where they have a similar phenotype and see if we could find new genes. And the exciting thing here is it looks like we have found a new gene. So something like 16 different cases, 10 families and 6 singletons, are carriers of rare deleterious variants in TBX4. I show here the sort of families by family ID with the variants and the inheritance pattern. You see very importantly, the GHS allele frequency is the frequency that we've seen in our database with Geisinger. So these are variants that are not common at all in large numbers of individuals, and so because they're segregating in this way, we think they're really causative of PAH. So here's the full spectrum of variants from that study. You can see there's these genes that have been previously characterized as being associated with PAH, like BMPR2, which accounts for something like 13% of the cohort, but really exciting, about 10% of the cohort. Really the second hit for us is this TBX4 gene, which is new. So now reaching past the individual families, I want to talk a little bit about this kind of middle ground, places where you have people who are not unrelated, and we're not talking about large, case control studies of unrelateds, but things where we have kind of extended pedigrees and extended families. And one thing to think about is the way that we can infer a family or a pedigree from genotype data. So what one generally does is one looks at identity by descent. And the easiest way to think of this is the measure of the fraction of the genome where you share zero identity, that is, 0 of your 2 chromosomes are the same between you and the individual you're being compared to. IBD1 measures the fraction of your genome where 1 of the 2 chromosomes is shared between you or between the 2 individuals that are being compared. And so in this first line, talking about monozygotic twins or clones, having 100% IBD2, namely 100% of the genome, that is both copies of the chromosomes, are shared entirely between the 2. So for parental offspring relationships, on average, you have IBD1, that is, you get on average exactly 1 of your chromosomes from one parent and 1 chromosome from another parent. For full siblings, it's 25, 50, 25, et cetera, as you go into more distant relationships, all the way to unrelateds where you have 100% IBD0, that is, on average, you share no genetic material or no alleles between you or between the 2 individuals being compared. So this is a plot from a study we're working on with Toronto sick kids, where we've, like with the Mendelian disease program, like the previous study that I showed from Columbia, we've collected samples into families. And so what one expects is a lot of relationship here, because these samples were collected intentionally for relationships. So out of a relatively small number of samples, we have 13 monozygotic twins, you see in the lower left corner. In the upper left, at 100% IBD1, which is on the y-axis, and 0% IBD0 on the x-axis, you have parent-child relationships, and then on down that diagonal towards 2nd degree and 3rd degree, and you can see the cluster of full sibling relationships where you have 25% IBD0 and 50% IBD1. Now this is where it gets very interesting. This is, again, a cohort that was collected intentionally in families because we're trying to do a similar study in very early onset irritable bowel syndrome, similar to what we did in PAH with Columbia. But here's the Geisinger data set. So this is data from roughly 50,000 people. But remember, these people were not collected because they were related. These people were collected as part of the Geisinger cohort, collected because they're part of this healthcare system. And the thing that was very exciting and new to me is this data set is incredibly related. There are an enormous number, 4,900 full sibling relationships, 6,600 parent-child relationships. Remember, each of these points is a pair of individuals. And so at the end of the day, what we found was there were over 6,000 family networks or pedigrees within the 50,000 Geisinger samples we had sequenced. And you see the plot on the right shows when we collect samples into families, the number of samples in each family. And there are enormous numbers of families where there are more than 5 individuals who have been collected and sequenced as part of Geisinger, and that lets us reconstruct something like this, which I think is really cool, which is really only possible in a really exciting data set like the Geisinger data set, where each of the gray boxes that can be a little bit hard to see, let me see if I can help you out there. So there's a gray box sort of here, sorry, here, that contains all of the familial and sibling relationships, the parent-child and the sibling relationships. So you can think of these as nuclear families. These are, the gray boxes contain red lines and yellow lines that are connecting points, which are individuals who share those very close relationships. And then the blue lines are connecting 2nd degree relationships. These are cousins, these are aunts, these are uncles. And what you see here is over 1,100 people collected into a network of second-degree relationships where any single individual within this population is connected to another individual through this population through an identifiable familial relationship. So this then really is that middle ground where we aren't talking anymore about families, but we're also not talking about unrelated individuals. We're talking about really extended networks of relationship. And that enables discoveries like this. So this is a meeting of the minds between what we have done in copy number variation, where we identified a novel copy number variant, a duplication in an LDL receptor gene that basically takes the end of the gene and duplicates it. And you can see at the top sort of an ideogram of the gene, and then just below that, what we've done is we've actually done whole genome sequencing to reassemble that region and validate that this is a tandem duplication showing the microhomology at the breakpoint. But getting back to the families, the carriers of this variant, which was found doing an association between various cardiovascular phenotypes and various variants that we found, this variant actually collects in a large extended family. So all of the carriers that we have of this variant have a common ancestor 5 or 6 generations ago, and the carriers of this variant also are enriched for having ischemic heart disease. So what we found is essentially a familial variant. This is clearly a rare variant. We haven't seen it in other populations, although it is a copy number variant, and those can be hard to characterize, so it might be under ascertained. But at least in this population, if you have this variant, it is indicative of being part of this extended family, and it also is likely to have this functional impact of giving you higher risk of heart disease. Now, moving on, and I want to finish up so we have time for questions, so I'll just kind of point out that there's a variety, again, moving over to the left, of more conventional kind of case control studies that we can do. So, and we've published on a couple of these now, so you can go look up the papers. So in this example, what we've looked at is HPTL3, and we found that loss-of-function variants in HPTL3 are associated with reduced risk, with reduced cardiac disease risk, as well as reduced lipid fraction. And the interesting thing is we have a drug in development that is targeting this gene. And so this is really driving, this is a great example of how the genetics can drive our drug development efforts. We have a drug in development, we can look at the gene, we can look at loss of function carriers, we can do statistical association studies, identify the fact that there's a very strong association between total cholesterol and loss of function carriers for this particular gene. Here's another interesting example. This is an HPTL4, and this there's a reference here, so you can go look up the paper if you're interested in learning more. But really cool about the HPTL4 story is one of the variants that you see here, the E40K variant, which is quite common, doing a large-scale study, doing a replication of a discovery that we found in about 30,000 individuals within Geisinger, where there seemed to be a reduced risk of type 2 diabetes among the carriers of this variant of HPTL4. That variant validated doing a combined study across 300,000 individuals from a variety of studies. So again, this hits to the theme of collaboration. We really need to all work together, particularly in these replications, so we can understand the robustness of the signals that we're seeing in smaller studies, but it gets really exciting when you start to see that this is a really interesting result where we can start to think about what it would look like to try to use this lever to reduce risk of diabetes. And I would be remiss if I didn't show at least a little bit of functional data. So Regeneron has a great history of working in mice. So here's an example where we've looked at some HPTL4 deficient mice, and we've shown in terms of blood glucose, in terms of glucose and insulin tolerance tests in mice on a high-fat diet, that glycemic control is better in the mice that are HPTL4 deficient. So that is a nice functional validation of it, and starts really setting the mind about what might be possible with some of these association results. All right, so I'm going to stop there. I do want to say just a quick word in terms of conclusion in the future. So I like to say, the cloud isn't the future, the cloud is now. There's a lot of talk that has been had both in enterprise IT, research IT, and the research community over the last 5 years about the cloud. For us, I can tell you our story, I can't speak to everybody's situation, but for us, it's really proven to have lower costs, be more flexible, be more secure and more scalable than local hardware. And one of the really exciting things for me in particular, because again, hitting that theme of collaboration, this really enables frictionless collaboration. We can have shared data areas with partners that are in other states, in other countries. All they need is an internet connection. They don't have to have their own high throughput computing facility. And also enables frictionless cross-collaboration. When we have a collaborator who finds something interesting and maybe that creates a natural scientific connection with another collaborator, they can connect with each other, they can connect with the data in the cloud in this secure way. And I think this is really, in some ways, the secret sauce to the future of genetics. In terms of genomic-driven drug discovery, this is absolutely the now for us. You know, Mendelian disease, you saw this great PAH example, offers really clear insights into the biological impact of high-impact LOFs. More subtle effects can be achieved with proper case definition and large cohort analyses. Some of you know, the exciting results you saw in HPTLs. And of course, these are not new ideas. But the sequencing compute technology and the kind of collaborative perspective that we have, I think, are unique enablers for us. And then let me just end with collaboration really is the future, but it's also the now. This is something I think that large-scale genetics has reached for a long time. We're going to need more and more of this if we really want to change medicine, if we really want to change science. We have to innovate all at the same time in biology, in medicine, in computation, genetics, drug development, data science. So we have to get it done. We really need to have these multi-type cross-collaborative efforts, which is a nice segue into the acknowledgements. The RGC leadership team, particularly our collaborators, David Carey, David Ledbetter, Marilyn Ritchie at Geisinger, Wendy Chung at Columbia, and Omar George Mike and Ohad at DNA Nexus, as well as, in particular, my team, most specifically John Penn working on production, Lucas Haybecker taking responsibility for R&D, and Chris Sprengel and Rick Lowe working on IT and LIMS, all of which has to work together to make this work. So thanks for listening, and I would love to take whatever questions you have.
All right, well, thank you, Jeff, for that captivating and truly inspirational presentation. It's an absolute tour de force. It's time for Q&A, so out there, if you have any questions you want to ask Jeff, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many questions as we have time for. And let's see. All right. All right, well, here's one. Jeff, why did you decide to use your own CNV caller?
Thanks. No, that's a great question. So what we did was, honestly, we looked at the tools that were out there, and we could not find a tool that would call common copy number variants, call rare copy number variants on exome data, and do it in a scalable way. And in fact, we really could only find, most of the tools that were available that were working on exome data were really focused on doing rare copy number calling in small data sets, and so they just were not suited to this purpose. I think part of it is other people haven't had data that has been made as uniformly and as consistently as our data. You know, historically, larger genetic studies, you know, have been made over longer time periods because we started at a time when we could make data sort of faster and more cleanly than people had been able to do years ago. And so, you know, we had some unique data set. We had some unique interest in doing both these rare studies and these common variant studies. And so for us, it really wasn't a choice. If we wanted to do copy number calling from exome data for common and rare variants across 100,000 people, we were going to have to develop something new. And I'm just really proud that the team was able to actually deliver on that goal.
Well, given the quality of the team, I'm not very surprised. All right, another one's coming up. What was the motivation behind committing to the cloud from the beginning?
So there were actually a variety of similar motivations. So there wasn't just one thing. But first and foremost, you know, things at Regeneron move pretty fast. We have an idea and we want to execute on it now. And so for us to build a physical infrastructure, a physical compute infrastructure that would be needed for the scale of sequencing that we needed would just have not fit well with the timelines that we were tasked with. So we knew we needed to stand something up really, really fast, but we also knew we needed to stand something up that was really scalable because at Regeneron, not only do we do things fast, but we do things big. And so we also knew that once we were able to produce sequence, that it was almost certain that we would be tasked with producing even more sequence. And that absolutely happened. Within months of really starting sequencing at the RGC, our goals were doubled. And there was just no way that we would have been able to, one, put together the money and the effort to build it physically, and two, scale it to our needs. And, you know, so we looked around at what solutions were out there, and for us, it was very clear that AWS and DNA Nexus was a solution that would really get us what we needed and where we needed to go, and the team has been incredibly helpful in working with us, and it's really fun and it's really collaborative working with those guys.
Okay. See on time. Okay, how can I get involved in this kind of research?
So it depends on, I guess, what I'm not sure what the question means by how do I get involved, but I'll sort of ask the question two ways and answer it two ways. So one way of asking that question is like, how can I get involved in this research as a study participant? And I think one of the very interesting things that has emerged is the Precision Medicine Initiative. And I know that they have a variety of efforts, and they're really trying to incentivize participation from everybody. And they're very interested in getting people who are willing to you know, give over DNA, give over health record data, and really, you know, participate in the study as somebody who's giving you know, of themselves to try to help science. And I think that's really laudable, and that's really exciting, because in the last 5 or so 10 years, it's maybe not always been so clear if you really wanted to participate in something like this, how you could. The other way to ask that question that I think is also really valuable to think about is, you know, as a scientist, as somebody who's interested in medicine, as somebody who wants to see healthcare get better, you know, how do I participate from the research side, not as a participant, but as somebody who's doing the work? And there, you know, I would just say that I think this is an incredible field. It's growing just leaps and bounds. You see more of this kind of sequencing every day. You see more aspirations towards precision medicine all over the world every day. And, you know, I would say getting an education where you understand fundamentally genetics and biology, but where you also fundamentally understand computing and data science. I think people with those skills will be very well positioned if they want to get involved in this kind of research from the research side in the future. And then, you know, the other thing is, just look around in your local area. This is really happening everywhere. That's one of the reasons I think it's very exciting. And so, you know, go to your local institution of higher education, your local healthcare system, your local hospital, your local, you know, healthcare industry center of excellence, and I bet you'll find some of this. And, you know, if you're really, really motivated, I have no doubt that everybody can participate and contribute to this.
All right, so remember, kids, if you're out there listening, Jeff Reid says, stay in school and learn genetics. Since we're just about out of time, I'm going to take the moderator's perspective because I can't resist and ask Jeff this final question, a kind of a pet topic. Everyone talks about the promise of geno-pheno integration, and certainly, you know, at DNA Nexus, we're driven and keen to catalyze this. From your talk, it sounds like you're making remarkable, really concrete progress towards actionability, both on the drug development side, where it's absolutely remarkable. I've never seen anything like this, in terms of target ID, and then also in terms of actually being able to combine the information to have clinical impact, at Geisinger. From your perspective, how do you think we're doing on this journey? Where are we, and what have you sort of been most surprised or proud or excited about?
So it'll be hard to sort of pick one thing to be most excited or proud of. I think I'm quite proud of the RGC in general, in that it was not obvious that this kind of academic sequencing effort at a pharma company could or would or should succeed, and I think it has on all counts, and that's really gratifying. But to get more to the actionability, I think that the one thing that I think is really, really cool, because it's something that I've been thinking about for a while. There just isn't a lot of data that is coming back to patients about CNVs. There are a small number of CNV tests that are run. I think they tend to get run for people who have a particular phenotype or a particular disease area. And I think we're going to see very soon. I know Geisinger's been working really, really hard on this and trying to do it right. But I think we're going to see really, really soon some of the CNV data actually coming back to patients, getting some information about, that they're carrier of given CNVs, and those CNVs are thought to be associated with phenotypes that they have, and giving them kind of more information about their genotype and maybe help them get some insight into the phenotype as well. That, to me, that aspect is particularly gratifying, but just generally as well, the idea that this data is, ending up back in the hands of clinicians who are, doing validation and thinking about it rigorously and putting it together into reports that are going to be impactful and helpful to patients, and getting data back to the people who are putting samples in there. Not every study gets to see that, and so that is just really, really exciting.
All right. Well, it's clear that you're, though not located in Silicon Valley, nevertheless achieving the Silicon Valley ambition of making the world a better place. So we appreciate that. I really want to thank you for your absolutely spectacular presentation, and also to thank our sponsor, DNA Nexus, for making today's educational webcast possible. Do you have any final parting comments, Jeff?
I do. I just, thanks everybody for sticking around and listening to this for so long. Obviously everybody has a lot going on in their day. I think the things that we're doing are really exciting. I hope other people are excited and motivated by them too, to go and do similarly exciting things, because, the rising tide floats all boats, and we really want to see everybody getting their hands in this data and doing, genetics in this exciting new way. Because if we really want to push this field forward, it really does, hitting that theme of collaboration again. It's really going to take a lot of different people with a lot of different backgrounds working together. So I look forward to working with some of you and hearing from great things from many or hopefully all of you in the future. And thanks again for listening.
All right. Well, thank you so much, Jeff. Really, just incredible stuff. Before we go, I do want to let everyone know that today's webcast will be available for on-demand viewing for 6 months from today. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues or anyone else who may have missed today's live event. All right. Well, that's all for now. Thank you for joining us, and we hope to see you again soon. Take care.